this video, we will be talking about point mutations. We will go over missense, silent, nonsense, and read-through mutations. First, we will talk about transition versus transversion mutations. Transition mutations occur when a purine mutates to another purine, or a pyrimidine mutates to another pyrimidine. This occurs when an A or a G mutates to a C or a U or a T, or the other way around. However, transversion is when pyrimidine mutates to a purine, or a purine to a pyrimidine. For example, when an A or a G mutates to a UT or a C, or the other way around. Now to define the mutations. Missense mutations changes the amino acid such as altering the protein or making it non-functional. However, it can be neutral, where the mutation doesn't affect the protein's function. Nonsense mutation is a change to a codon to a stop codon. Silent mutation is when the DNA is changed, but it still codes for the same amino acid. Read-through codon changes the stop codon to amino acid codon, making it into a longer protein. Before looking at the mutations, we must transcribe this DNA code to mRNA. We will replace the thymines with uracils. Reading from 5' to 3', the start codon will be AUG, which is 3 letters long, like every codon. We go through the strand, identifying the other amino acids until we reach the stop codon, which will be UGA in this case. Now the mutations with the given strand. Missense mutations. Let's change the AAC to AUC. The change from A to a U, it is a transversion mutation. Also, the protein would change from a asparagine to a isoleucine. Next, the silent mutation. Let's mutate the GCU to a GCG. The transversion occurs when it changes the T/U to a G. The protein alanine remains the same. This is why it is silent. Now, let's look at a nonsense mutation. Mutation changes from UGG to a UGA, which is a stop codon. Therefore, a nonsense mutation, the G to a A base pair, is a transition. Lastly, read-through mutation. The original stop codon AUG, which mutates for UGU, which is a cytosine, that means you don't stop there. Therefore, this is known as a read-through mutation.